I'm Connor Kramer.
I'm Michael Payton, and this is Period 3.
The overall purpose of this lab was to cause a mutation in a gene, BLI1, and be able to observe the change in the worm's phenotype. This was done by exposing C. elegans to RNAi bacteria, which they consumed on a plate. This causes a decrease in the expression of the number of BLI1 mRNA in the cells of the C. elegans, which essentially turns off the gene by inhibiting the amount of mRNA that can be translated into a protein. But before we begin the lab, we must learn the sterile lab procedures. Connor.
First, you're going to want to spray yourself down with 70% alcohol to keep yourself from infecting your lab. Make sure to rub it all around to get it all over your body, that way you're not infecting anything. Next, you're going to go into the biological safety cabinet. Once your hands are in, don't take them out or else you're going to infect your lab. Make sure you're sitting in a seat so that your arms are in a comfortable position. You're going to want to start by grabbing your spatula and dunking it into the alcohol. Once you've done that, put it over the open flame and let it cool for 5 to 10 seconds. Once that is done, pull your petri dish over with one finger and open it up. This one has the auger. You're going to want to cut a small square out of it and pick it up with the spatula. Once you've done that, close your dish and move it away and bring the feeder plate over. Open the feeder plate and make sure when you're opening the petri dishes to keep the lid over it to keep it from contaminating. Flip over the spatula and lay it down nicely in the corner and close it and push it away. You're then going to want to dunk it in the alcohol and burn it again to disinfect it, and then you're done.
Here you see the wild-type C. elegans worms. They move along in an S-shaped motion in the plate. Occasionally you will see their heads shake slightly. This indicates that they are feeding on the bacteria that is in the plate. They are generally clear, however will appear slightly darker depending on how they're lit under the microscope or how far along they are in the adult stage. Their life cycle starts out with the egg, seen here, then they hatch and progress through the L1, L2, L3, L4, and finally mature into the adult stage seen here. Here are the mutated C. elegans worms. As you can see, the silencing of the BLI1 gene causes these large blisters to form on the bodies of the adult worms. They only start to form during the adult stage in their life cycle and will eventually lead to the adult's death. These blisters interrupt their movement and in some cases cover the entire body of the worm.
The BLI1 gene codes for cuticle collagen proteins, working with others, including BLI4, which are responsible for proper strut formation in the medial layer of the cuticle in C. elegans. BLI1 mRNA is highly expressed in the adult stage, which is why when it is silenced, when the worms reach the adult stage, they develop blisters and perish.
Some of you are wondering what the process is behind this experiment. What causes a worm that looks like this to mutate into a worm that looks like this? Well, the process begins with the worm eating a cell on the feeder plate that produces double-stranded RNA, or dsRNA for simplification. The dsRNA then enters the cell and triggers the RNAi pathway. Dicer then chops this dsRNA into smaller segments called small interfering RNA, or siRNA for further simplification. Then this siRNA binds to other proteins producing RNA-induced silencing complex, or RISC. Then what's left of the double-stranded siRNA is unzipped and leaving a single strand of mRNA. RISC then probes for mRNA in the cell that matches the mRNA that we put into the cell. It then binds to the mRNA that the cell produces and cleaves it, effectively stopping the mRNA from transcribing into any protein, therefore silencing the gene and turning the worm into this.
The results we observed did support the function because we saw blisters on our C. elegans. Overall, our group really enjoyed this experience working with C. elegans. We learned how to work with model organisms and how to modify the C. elegans. This is Connor, Mike, and Braden signing off.